Dear students, today let us discuss about Megasporogenesis and female Gametophyte, which is a very important topic in our embryology. So in order to study this Megasporogenesis, the definition of Megasporogenesis is formation of the megaspore. During the development of this megaspore, megaspores are developed in the female organ of a flower. So in order to know all these things, we have to go a bit earlier of this part of embryology. We have to discuss about the gynoecium. What is gynoecium? Gynoecium, it is the female reproductive organ of a plant. In a plant, there may be the gynoecium, which is the female reproductive organ, and both the female reproductive organ and the male reproductive organ may present in a flower also. For the time being, we are going to discuss about the Megasporogenesis, which is concerned on the female part. So let me discuss about the gynoecium. What is gynoecium now? Gynoecium is the most commonly used term it is the collective term for the parts of a flower that produce ovules and ultimately develop into fruits and seeds. What are the parts? The parts are the carpel. It may be also present in the female reproductive organ gynoecium. The carpel is the main component of gynoecium that consists of ovary, style, and stigma. The end of the carpel receives pollen grains is called stigma. It is a long, narrow, tubular structure, and it is present in between the stigma and ovary called style. So the vessel solent part of the carpel is called ovary. Then ovule or the megasporogium are also present here. Then what is ovule now? Let's discuss. The ovule is also known as the megasporogia, which are borne on the cushion-like tissue called the placenta in the ovary. Reeds or stalk like outgrowth is formed from the placenta of the ovary, on which body of the ovules are present. So in each ovule, there is the attachment to the placenta by means of a thin stalk called funicle or funiculus or funiculum. So the point of attachment of the funicle with the ovule is called hilum. What are the main reasons of the ovule? So the main reason of the ovule is composed of mass parenchymatous cells called the nucellus. The nucellus is the main part of the ovule. The nucellus is again covered by one or two coats called the integuments. In most of the ovule, funicle is attached to the main body of the ovule for some distance per lateral side to form a ridge-like structure known as the raphe. Now in this structure, this is the structure of a mature ovule and a mature embryo sac. In the mature ovule, what are there? Nucellus is there, salazal end is also there, microphyle end is also there. So what is there in the embryo sac? Embryo sac, it is a 8-cell structure which we are going to study here. There is the antipodal cell, 3 antipodal cell is there, 2 polar nuclei is there, and the synocytes and the egg cell is also there. So what is the explanation of all these things? A place from where funicle and integuments arise is called chalaza. So integument is absent in just opposite to the chalaza, so that the narrow passage or the pore is formed, which is called microphyle. In most of the angiosperms, entire part of the nucellus is utilized by the developing embryo sac. So the tissues present in the embryo sac, they are utilized by the angiospermic plants, but in some of the angiosperms, some part of the nucellus may remain inside the ovules. That part of the nucellus present inside the sac in the form of a thin layer known as the perisperm. It is commonly found in some parts or in some family, name Piperaceae, and namely maybe the Piperaceae and the Zingiberaceae also. So according to the what types of the integuments and how many integuments are present, based on these integuments, there are two types of the ovules. These two types of the ovule is, number one is the, as the name indicates itself, it is the unitechmic ovule. Uni means the one. So a single integumented ovule is called unitechmic ovule. For example, it is found in the Gametophyte and the gymnosperms also. And the second one is the bitechmic. The literal meaning of the bi is two, so there must be two integumented ovule, and these two integumented ovule is called bitechmic ovule. Example, it is found in angiosperm, and in the endosperm also the families like the polypetalae and monogoch, this bitechmic ovule is found. So these are the points based on the integuments. Now let's study types of the ovules are also again on the basis of the nucellus. There may be the type of the ovule also. So the type of the ovules which are based on the nucellus, these are the crassinucellate. Crassinucellate is the nucellus is massive. It is made up of many layers, and the sporogenous tissue is deeply embedded in it. Such ovules are known as the crassinucellate ovule. And another type is the tenunucellus. It is unlike that of the crassinucellus. The nucellus is either less developed or present in the form of a single layer around the sporogenous cell, such as the ovule, and it is called the tenunucellus. So on basis of the nucellus also, these ovules may be two types. First is on the basis of the integument, and second is on the basis of the nucellus. Now there are many types of the ovules are also. It is based on the position of the microphyll and chalaza at maturity, how this microphyll and the chalaza are arranged in a form of the ovules. These ovules are of many types. The following are the types. Number one is the atrophous or autotrophous ovule. These two terms are the synonymous terms. So atrophous or the autotrophous ovules, you should be clear about these two synonymous ovules. Atrophous is another name of autotrophous ovule. So the body of the ovule is upright here with the microphyll, chalaza and funicle. They are falling in the straight line, so that this ovule is called straight or upright ovule. For example, this type of the ovules are found very commonly in the Polygonaceae, Piperaceae and gymnosperms, and it is the most common type of the ovule also. Number two, it is most primitive and the most simple type of the ovule of the angiosperm. So these are the 2 points. The ovules, the microphyll, chalaza and the funicle, they should be in a straight line. And another one is, it is the simplest type and the most common one. Second one is our anatrophous ovule. What is anatrophous ovule now? How this microphyll, chalaza and the funicle, they are arranged in this type. Let's discuss. Due to unilateral growth of funicle, the whole body of the ovule is inverted to 180 degree. As a result, microphyll come close to the base of the funiculus. Due to unilateral growth of the funiculus, so it is also called the inverted ovule. The nucellus remains straight, microphyll and chalaza lie in one line, and the funicle lie parallel to it. What is the difference between the first one and second one? In the first one they lie straight, but in the second one the nucellus remains straight. That is, the microphyll and chalaza lie in one line and the funicle lie on the parallel of it. So it is most common type of the ovule and occurs in several families of both dicotyledons and monocotyledons. Now let's discuss the third one. The third one is the hemitrophous or hemianatrophous ovule. Sometimes you may find it in some books as the hemianatrophous ovule. It is same. So in this ovule, the body of the ovule bend on funicle at 90 degree angle. That is, body of the ovule present at right angle to the funiculus. This is intermediate type between the ortho and the anatrophous ovule, which we already discussed previously. So this ovule is also called horizontal ovule because the body of the ovules present in the horizontal position is on the funiculus, microphyll and zalaza are present in the same line, but the microphyll is situated away from the hilum. And how this happens, also it is 90-degree angle is bent. And the example of this hemitropous ovule is found in the families like the Ranunculaceae and Primulaceae. This is the hemitropous ovule. The fourth one is the campylotropous ovule. This ovule, the body of the ovule is curved in this way. Microphyll and zalaza do not present in the straight line. The embryo sac and nucellus both are present in curved position. Microphyll comes close to the hilum, and this type of the campylotropous ovule is found in the Leguminosae, Caparidaceae and the Cruciferous family. What is the Cruciferous family? Cruciferous is your mustard family, Brassica family. So in most common type of the Brassica family, this type of the campylotropous ovule is found. The fifth one is the amphitropous ovule. How in the amphitropous ovule, how this microphyll is arranged with the nucellus and funicle. In this type of the ovule, curvature is more pronounced or effective in the nucellus, and due to this effect of nucellus, embryo sac becomes horseshoe shape, like in the picture. So in the number 2 point, this microphyll comes close to the hilum. How this microphyll comes close to the hilum? It is so like that. Usually microphyll should be straight in the upper side, but due to the curve or due to the angle of curvature, it have become close to the hilum, which is present in the funicle. It is also called transverse ovule. It is found in some families like the Butomaceae, and the plants which are present in this amphitropous ovule are the Mirabilis, Lamna and Phoebe, like this. So the amphitropous ovule, in the amphitropous ovule, it is horseshoe shape, and it is more pronounced and effective in the nucellus. The sixth one is the Sarcinotrophous ovule. Sarcinotrophous ovule, it is very different type from the previous 5 which we discussed. And the sixth one is the Sarcinotrophous ovule. Here, this type of ovule, in this type of the ovule, first of all, body of the ovule is totally inverted and once and again turn into straight position due to the growth of funiculus, so that body of the ovule present on funicle at 360 degree. It turns to 360 degrees, so it is totally inverted. The entire body of the ovule is surrounded by funiculus. It is also known as the coiled ovule. Microphyll is situated away from the hilum. Example, it is found in the Cactaceae, and in the Cactaceae family also in the Opuntia, which is very common. We can see it everywhere. And in this Opuntia, this type of the Sarcinotrophous ovule, which is very specific one, it is present. Now, what is the Megasporogenesis? Come to the point, Megasporogenesis and the Megagametogenesis also there. This Megasporogenesis is nothing. It is the formation of the megaspore from the megasporangium. That is from the megaspore mother cell. In the megaspore mother cell, it is the twice end, it is the diploid, but during the formation of the megaspore, due to the meiosis, the chromosome numbers should be reduced to half, and it will become haploid end, and this megasporogenesis is nothing. It is the formation of the megaspore during this development. After that, megagametogenesis will also be there. This megasporogenesis, let's explain this megasporogenesis. About the megasporogenesis, I have already told you, it is the formation of N, it is the haploid from the megaspore mother cell, from the diploid megaspore mother cell, how this formation of the megaspore N takes place inside the megasporangium. What is the megasporangium now? It is the ovule, nothing, it is ovule, and the whole process is known as the megasporogenesis. How it happens? Now let's just discuss it. First of all, a single hypodermal cell in the nucellus is differentiated from the rest of the cells by its dense cytoplasm and a larger size and a larger nucleus. It will be distinct one. A single hypodermal cell should be distinct one, and this is called the primary archesporal cell. Once again, it is the primary archesporal cell or the archesporal initial. It is the first one, so it is also known as the archesporal initial. This archesporium divides periclinally to form an outer primary parietal cell and inner primary sporogenous cell. Activity of primary parietal cell depends on the types of plants, and if the plant belongs to Gametophyte, then it forms tenuine nucellus. We already discussed what is this tenuine nucellus type of the ovule, and if the plant belongs to Polypetale, then it forms crassy nucellus type of the ovule. The primary sporogenous cell directly acts as a megaspore mother cell. Megaspore mother cell, what is the chromosome number? It is diploid. So it divides meiotically. Meiotically, if we divide meiotically in a plant, what will happen? The chromosome number will be reduced to n, or reduced to half meiotically to form 4 haploid megaspore. The 4 haploid megaspores generally arranged in a linear tetrad. Generally, the lowermost of the salazar megaspore remains functional, and out of the tetrad of megaspore, the other 3 lie towards the microphyle and the degenerate. Like it in this picture, it is all shown here. This primary archesporal cell is also there. This primary parietal cell is there. Primary sporogenous cell is there, and how it divides is shown in the picture. Now, the functional megaspore produces female gametophyte. In most of the angiosperms, salazar megaspore remains functional. Now, this female gametophyte or the embryo sac, how this female gametophyte or the embryo sac develop. This process is the megagametogenesis. Like that of the Megasporogenesis, in the megagametogenesis, there is the formation of the female gametophyte, or the another name is the, you should not confuse, these female gametophyte are nothing but it is the embryo sac. So in the embryo formation of the embryo sac, what is the first cell of for the formation of the embryo sac? It is the megaspore. Megaspores are formed from the megaspore mother cell. If the megaspore are developed from the megaspore mother cell, then it is called the megasporogenesis. If the megaspore is the first cell and the embryo sacs are formed from the megaspore, then it is known as the megagametogenesis. Megaspore is the first cell of the female gametophyte. This megaspore grows in size and obtains nutrition from the nucellus. How this megaspores grows? It gets its nutrition from the nucellus. You should know it. It is very you should know this very carefully, how this megaspore gets their nutrition. So the nucleus of the megaspore divides mitotically to form 2 nuclei. Mitotically, if a cell divides mitotically, it should form 2 nuclei only. If meiotically it divides, it should be 4 cells. But here it is mitotically divided, so only 2 nuclei will be formed. Its nucleus moves towards the opposite pole and rests at their respective poles. Both the nuclei lie at poles, divide twice mitotically, resulting 4 nuclei are formed at its. Total how many? 8 nuclei is there. Inside the cell, there should be 8 nuclei. So in a female gametophyte or the embryo sac, generally it is 8 nuclei. That many nuclei type may be also present. We will discuss it later. But for the time being, there is only 8 nuclei in a normal type or the general type of this embryo sac. So out of the 4, one nucleus migrates from both the poles, 1 nucleus from the salazar end and 1 nucleus from the microphylar side towards the center. They are known as the polar nuclei. Both the polar nuclei are present in the center. Both of them are N and N. Later there will be fuse. We will discuss it later again. What happens now? Remaining the 3 nuclei at each pole surrounded by cytoplasm to form cells as a result of cytokinesis. 3 cells are formed towards the microphyle, in which one cell is large and more distinct out of the 3 cells. This is called the egg cell. Remaining 2 smaller cells are known as the synages. This egg cell is present in between the 2 synages. These 3 microphylar cells collectively known as the egg apparatus. One egg cell will be there and the 2 synages guarded by the 2 synages. It will form a cushion-like shape for the egg cell. The 3 cells are formed towards the salazar are called the antipodal cells. The function of the antipodal cells also we will discuss later. Both the polar nuclei are present in the central cell, but just before the process of fertilization, they unite or fuse together in the center to form secondary nucleus. It is diploid in nature and 1 in number. Diploid in nature because N and N are fused, and if they have become 1, there will be the diploid in nature. Therefore, 7 cells and the 8 nucleated structure is formed here. One in the polar nuclei, they are fused to 1 cell, so now it has become the 7 cell, but the nucleus is 8, so it has become 7 cell and 8 nucleated structure. This 8 nucleated and 7 cell structure is called female gametophyte or the embryo sac of angiosperms. This type of embryo sac is known as the polygonum type. Then now, this polygonum type of the embryo sac is the most common one and general one. It is very universal one. And the finger-like processes are produced from the outer wall of the synages is known as the filiform apparatus. With the help of this structure, synages absorb food from the nucellus. That this nucellus is the food of this developing embryo. So this synages absorb food from this nucellus again and transfer to the embryo sac. Filiform apparatus is less developed in antipodal cells. Filiform also secrete chemicals which attracts the follicle. These are the development of your megasporogenesis and the female embryo. So students, today let's just end of our topic. In the next topic, we will study more about your embryology.